evening everyone. Welcome you all to week 12 NPTEL live session of RNA biology. Today we are going to discuss few questions on RNA epi transcriptor and translation mechanism. Okay. Earlier we have been discussing about telomerase enzyme and how telomeres are helpful in increasing the lifespan of a particular cell. Okay, so this is Subhasmita. I am a PhD scholar at IIT Indore. So today those questions will help you to understand the assignments and complete your assignments correctly. Fine. So I think we should wait for 2 to 3 minutes more, so that more people can join. So we'll start the class from 6:10. Okay. After 3 minutes we will start the class. Just wait for 3 minutes. Okay, let's start. So, in previous class, we have been discussing about uh, telomerase, okay, telomerase enzyme and about the telomeres. So, we will just recap it in just a few minutes, okay. So, at first, what is telomeres? So, telomeres are the repetitive sequences, okay, they are the repetitive sequences which we can find at the end of a chromosome. So, why are they present there? They are present at the end of chromosome to protect it from interacting with the other end of the chromosome, also from exonucleases. Okay, this is one function of your telomere repeats. Got it? Next is, what is its function again? It helps in the stem cells and germ cells to lose any genetic information through DNA replication process. Okay, let's see how does it work. So if you see here, this is the strand of DNA that is in 5' to 3' direction. Another strand is from 3' to 5' direction. Okay, so for example, this is your origin of replication. Got it? So which can any position of a DNA can be a origin of replication, and the replication process starts from there only. Fine. So this is whole thing is called replication bubble. Okay, so if you see here, you can find out that from in the 3' to 5' strand, DNA replication happens in a continuous manner. So this is called a leading strand. Okay, this one is called leading strand. But in the 3 days to 5 days template strand, this DNA replication is occurring in a discontinuous manner with producing different Okazaki fragments. So these are called lagging strand. Got it? So what happens if we just sum it up? So after the completion of replication, we will find out that there is some end RNA primers are there. So these red color things are called RNA primers. Okay, don't get confused. So these are the RNA primers. So these RNA primers will be get removed by DNA polymerase alpha. Fine. And in that place, this nucleotides get added and the DNA strand will be completed, and the other DNA strands can be ligated using the DNA ligase enzyme. I think everyone knows about this. First, then after removing the RNA primers, you can find out there is some space is there at the 5 days end of the both the DNA strands, right? There is some space. So this space, because of this space, we need a complete whole strand. So in that case, your other enzyme will cut this FBA1 will cut the overhang. So this is called overhang. Got it? This is the overhang. So here the enzyme will cut the overhang of the DNA so that we can get this much amount of new DNA. Understood? So this will be a pair of DNA that will be added as a chromosome. So in case of our somatic cells, this overhang get removed. So if you can see here, this overhang means it has some important genetic information which is going to lost by the cell after each and every replication process, right? That's why our somatic cells age or they move towards senescence. After a number of replication, a number of cell division, the cells move towards senescence. That's why in case of uh, older individual, the number of telomere repeats are very less as comparison to the infant ones. So here this genetic information which we are looking at that it is going to last, these are not genetic information, these are only for telomeric repeats. This can be of 10 to 12 kb in case of an infant, but after a multiple cell division process, the length of telomeric repeats decreases up to 1 to 2 kb. Okay, that's the case in case of older individuals. And when this telomeric repeat completely decreases or lost, then the original DNA gets destruction, move towards destruction, that will lead to production of malfunction proteins and RNA, so that the cell will die. Understood the whole mechanism, how these telomeric repeats are helping us to without losing any genetic information, we can get the whole copy of genetic information. Okay, so here comes our enzyme called telomerase enzyme. So what it does, this enzyme has important role. What it does, it generally adds the telomeric repeats to the end of chromosome. Okay, so how does it add? So this is one picture of telomerase enzyme. Okay, this is the protein part, this is shown in green color, and this strand you are looking at, this is the RNA template. Okay, so telomerase enzyme is basically a ribonucleoprotein complex. Fine. So this is basically this ribonucleoprotein complex. It has its own RNA and a protein molecule. Fine, which has catalytic function. So first thing, why do telomeres add these nucleotides at the end of chromosome? In case of germ cells, in case of stem cells, and other exceptions are there. So germ cells and stem cells, we do not want to age those cells. We do not want those cells to move towards senescence, as they are very important. Stem cells have pleiotropic property, they can give rise to multiple organisms. Also in case of germ cells, they are producing the sperm and ovum in different organisms, and they are essential that they should not move towards senescence. Okay, that's why here the telomerase enzyme adds the nucleotides or increase the length of telomeric repeats. How it does this? So here you can see this is the overhang. This part of your DNA or chromosome has been shown here. Okay, so this is your overhang. Generally we can cut this amount of overhang, which can be a telomeric repeat in case of somatic cells, and the length of telomeric repeats will decrease after one replication, other replication, other replication. But here in case of germ cells or stem cells, this telomerase binds to this 3 days overhang of the telomere, which is complementary to the telomerase RNA. How it becomes complementary? This telomeric RNA molecule, which is a single template, it just form a complementary interaction with at least 2 to 3 nucleotides, not much. Okay, only 2 to 3 nucleotides it forms the hydrogen bond with, that is means complementary strand. So now you can see this part is already left. So what happens? Using this CCAAUC sequence as the template, your DNA polymerase will add multiple nucleotides. Okay, now you can see the length of your upper new DNA is been increased. So like this way, this telomerase will shift towards little bit towards right, and after that it will increase its length much more. Okay, so now you can see in case of your new DNA strand, if you just cut the overhang like this much, you are getting the whole genetic sequence. Your telomerase is not going to reduced. Okay, so this CC sequence, this is here. So now this part of your telomeric repeats have been reduced. That means we are getting the conserved telomeric repeats as it is, as the telomerase enzyme just added the sequences, and those sequences were get destructed by your enzyme FBN1. Got it? So in case of telomerase stem cells and germ cells, they happen like this, so that the cell will not move towards senescence and it will be active all the time. Okay, but in case of some somatic cells, when there is some cancer and everything, some kind of inducers like tumor progression is there, these cancer cells produce their own telomerase enzyme. Okay, they produce their own telomerase enzyme and they activate the somatic cell so that it will not die, or that's why this is the mechanism how these cancer cells just surpass the death, cell death or apoptosis. Okay, we know that cancer cells just keep on growing and keep on moving towards different cell division phases, but they do not die. Why they do not die? Because of this, because of high telomerase activity. Okay, high telomerase activity is different. Telomerase activity is important in case of stem cell and germ cells. Okay, but high telomerase activity is observed in case of cancer cells. Okay, so for example, if you get some tissue from any part of a patient body, and you just found some high telomerase activity on that particular tissue, then you can say as a hallmark, telomerase activity is a hallmark of cancer. You can say that some tumor progression is going on there. Fine. So this is all about telomerase and telomeric repeats. Why is it very very important? Okay, and there are different medicines, drugs are available in some part of the country, which, and scientists have done many experiments where people can take this telomerase, keep on increasing the telomerase activity of the body so that the person can live longer. Okay, you can do some research on this also. Fine. So based on all the discussions we had just, so try to answer this question. Which one is called immortalizing enzyme? Telomerase, DNA polymerase, RNA polymerase, or none? Please write your answer in the chat box so that after 2 minutes we can discuss about it. Okay, let's see. So this is your answer, that is telomerase enzyme can immortalize the cell, and as per the study published in Journal of Science, the human retinal epithelial cells and foreskin fibroblasts were transfected with vectors encoding human telomerase catalytic subunit HTERT. Okay, so how this has been done, this is a study or experimentation was done by some group of researchers and published on the Journal of Science. So let's see what they did. They transfected the human retinal pigmental epithelial cell and foreskin fibroblasts were transfected with vectors encoding the human telomerase catalytic subunit. Fine. Next, the control cells showed the telomere shortening. Obviously, the control cells, like the somatic cell, generally will follow the telomere shortening, as well as normal levels of beta galactosidase as a marker of cellular senescence. So, when there is high telomerase activity, that is telomerase plus, transfected cells exhibited longer telomeres. What we have been just discussing about, that this happens, that happens, which this already just proved by a group of researchers. You can read this paper also. Longer telomeres and reduced level of beta galactosidase. So this beta galactosidase is a marker of cellular senescence. Fine. By the time the study was published, the high telomerase activity cells had exceeded their expected lifespan by 20 plus replication. Now you understand what is the function of high telomerase activity. Okay, so generally we can say that your germ cells and stem cells, they require a telomerase activity so that they can continue their living process and they will not move towards senescence. But when there is very high telomerase activity, that is a hallmark of cancer. Okay, and you and in our different cosmetic industry and different types of experimentation have been going on to increase the lifespan of human by different medications and all. So here, including the telomerase enzyme, you can do some experiments and experimentation and follow the research process. Okay, so here is the next question. Telomerase can restore youthful phenotypes in whole organism. Is it true or false? see. So some of you have said true and some of you have said false. So what is it? So this is also some experimentation has done on it, and here is the some kind of observations which we are going to discuss. The telomeres can restore youthful phenotypes in liver tissue. Okay, normal fibroblasts were transfected with HTRT, that is human telomerase activity enzyme, at, then grafted onto the mouse skin. Fine. Mice grafted with telomerase negative control cells exhibited phenotype signs of senescence. This is talking about the negative control. That means where there is no telomerase enzyme have been included. That means not grafted with telomerase enzyme. So the sign of senescence. That is the cell is moving increased fragility, reduced level of collagen 1 and 3, and subepidermal blistering. So what is reduced levels of collagen? For example, in old persons, all of us have seen that they have wrinkles, right? In the skin, wrinkles are very prominent in case of old persons. Why does that happen? Because of your lack of collagen, the skin loses. Okay, it cannot tighten as much as it was in the infant cell. That's why this is the example where the cell is moving towards senescence. There are some observations including the increase in fragility, reduced level of collagen 1 and 2, and subepidermal blistering. So these are the three different signs you can call that the skin cell is moving towards senescence. So mice grafted with telomerase plus cells, that means which has telomerase activity, high telomerase activity, exhibit a youthful phenotype despite the same number of replications. So even after the number of replications are same, this youthful phenotype have been observed with telomerase plus grafted mice. So what is youthful phenotype? So there will be no wrinkles, or there will be a good level of collagen present in that particular mice. So here this may arise to your mind that why cannot we do this so that our skin also will look like young, and every day in even in old time it will be like the younger ones, so it will be similar. So here also there are some medications and experimentation is going on, but there are different advantages and disadvantages are there, and some cases there are several challenges also we need to face to go through that particular process. Okay, and this telomerase can restore youthful phenotype in a whole organism also. So here we just talked about the liver tissue. Next is your a 2010 study in journal Nature showed telomerase activation in mice dramatically reversed the effects of aging. A restoration of youthful phenotype was seen in testis, spleen, liver, and intestines also. Okay, now you can see this intestine, which is very important when we are talking about gut microbiota, and gut is very essential for managing the micromanagement of the whole body. So here the intestines have also seen a youthful phenotype. Additional effects of telomerase activation was restoration of fertility, okay, reversal of cerebral atrophy, reactivation of neural progenitor cells. That means it helps your brain activation also. It helps to maintain the brain as active and young even in your old time. Fine. So this is the experimentation was done by the researchers, and here is our observations. So what is your answer? This is true. The telomerase can restore the youthful phenotype in whole organism. Okay. Fine. So here is another question of true and false. So exercise slows down telomerase activity. Is it true or false? for the previous question, this was the correct answer, that it is telomeres can restore youthful phenotypes in whole organism, and it is true. Okay, and for this question, all you can just answer it so that we can discuss. Okay, let's see. So, exercise increases the telomerase activity. How does it happen? So, um, this was when experimentation was done in mice model, also in human model. Okay, so in case of murine model, uh, voluntary running for 3 weeks. So this exercise induced, what was the effect of this exercise? A 2.9 fold increase in aortic telomerase activity. Aortic telomerase activity means you are talking about the heart muscle. Okay, so the telomerase activity of your heart, of the mice heart, just increased by 2.9 fold. So telomerase activity increased. What is the meaning of this? That means the cell is moving towards younger stage. Okay, it will not age suddenly or easily. Next, a 3.3 fold increase in telomerase activity in circulating mononuclear cells in the spleen. So why these cells in the spleen is important? Everyone knows that these are the places of producing your B cells and antibodies. Okay, eventually they produce the antibodies. So they are the place of activation of the B cells in the blood. And when your blood has much amount of antibody, so that it can fight against your foreign antigens. Okay, and this also 3.3 fold increase in telomerase activity have been observed. So here this is the thing we are talking about, a mice model. So what is the function what we observed in the human model? Compared to controls means those do not exercise, professional athletes exhibited a 2.5 fold increase in telomerase activity in young athletes. So that means the persons who exercise and work, or the athletes means obviously they are doing some kind of physical activity like running and every other things. So they just it the telomerase activity just increased 2.5 fold. That means the lifespan expectancy increases. Okay, that means generally people call or people tell that you should exercise and do some physical activity so that you will live longer, the number of diseases is going to happen to you would be less. Okay, so here this is also experimentally proven. So and other 1.8 fold increase in telomerase activity in middle aged athletes. So in middle aged athletes, this it has been found to be 1.8 fold increase in telomerase activity. So in both the cases you can see that increase in telomerase activity increases the lifespan of a cell. If your cell lives longer, that means the person is going to live longer. Okay, so if you want a disease free life with high life expectancy, then everyone should exercise and go for some physical activity except doing their regular work. Okay, so this has been experimentally proven, which have we generally hear from everyone. Fine. So this is false. Okay, Exercise slows down telomerase activity. This is false. Exercise increases the telomerase activity. Fine. Next question is, TA65 is purified from which plant? Astragalus, ashwagandha, asparagus, or none? Which one is it? If you have seen the lecture videos, then you know what is TA65. We are going to discuss about it, so don't worry. Just try to answer, or we can guess also. Okay, let's see. As you have said, astragalus, and it is true. Okay, so your answer is astragalus. So what is TA65? This is a naturally occurring highly purified single molecule derived from the Chinese herb astragalus. Okay, so earlier we have been talking about that telomerase activity just increased, and experimentation is going on so that people can take it as a medication, so to increase the life expectancy, which general people want. So this derived from a Chinese herb called astragalus. Okay, this activates the HTERT gene. HTERT gene has the human telomeric activity gene. Fine. And in vitro, it has been found, in vitro in mice model, it has been found that it moderately activated telomerase in human keratinocytes, fibroblast, and immune cells. Fine. Next is in vivo. It is administered as part of the patent protocol 1, with participant given 10 to 50 mg of TA65 per day for 12 months. Fine. So this is the medication which they provided, and TA65 is one example of your naturally occurring single molecule. There may be different artificial molecules are there in the market, or you can go for it and search for it and do some research if you are interested in this. But this is a naturally occurring, which is derived from a Chinese herb called astragalus. Okay, you can remember the plant name. Fine. So this is your another question. Which of the following is a frequent RNA modification? Deadenylation, ubiquitinylation, or N6-methyl adenylation, or sumylation? Which one is called has been seen as a frequent RNA modification? Okay, let's talk about it. So, which one is a frequent RNA modification? So, here it has been found that this N6 methyl adenosine modification is very frequent in case of RNA, as we are talking about RNA biology and how this RNA is impacting in case of different mutation and everything. Here is one example where RNA is get modified at the adenosine residue. Okay, it is methylated. This N6 methyl adenosine, that is M6A, okay, is an abundant modification in mRNA and DNA. It is a dynamic and reversible process and is the most pervasive modification in human mRNA. So why we are talking about mRNA? Because mRNA is the messenger RNA, which is very essential, produced by the transcription mechanism, and this mRNA will produce the protein molecule. Okay, so this mRNA has the codons which will be read and just move towards the move through by the ribosome and tRNA molecule to produce the protein molecule. So the genetic information is passed through mRNA, and if there is some modification in mRNA, that may lead to production of malfunction proteins also. It is found in some viruses and most eukaryotes, including mammals, insects, plants, and yeast also. Fine. It is also found in tRNA, rRNA, and small nuclear RNA, as well as several non-coding RNA such as XIST. Okay, so what is XIST? We just talked about XIST where when we are talking about the doses compensation. So SIXT and T6, they are just antagonists of each other. They are long non-coding RNAs, and they are responsible for condensing the extra X chromosome present inside a female organism. Okay, so this is about just a brief thing about XIST. The methylation of adenosine is directed by a large M6A methyl transferase complex. So how does this happen? So this methylation happen by a large M6A methyl transferase complex containing the MET-L3 and SAM binding subunit. So this is a S-adenosyl methionine, which is produced from the liver mechanism, and it acts as a methyl group donor. Okay, SAM is very important when there is a methylation process is going on. It acts as a methyl donor. So here is your methyl donor. Methyl transferase is your enzyme which modulates this process. Okay, so in vitro, in vitro means in mouse model, this methyl transferase complex preferentially methylates RNA oligonucleotides containing the GGACU sequence. So this is, there are some specific sequence which the methylation happen. So what is it? So GGACU, this is the adenosine nucleotide where this methylation happen. And a similar preference was identified in vivo, that is in cell petedosis. It mapped at M6A sites in raw sarcoma virus genomic RNA and bovine prolactin mRNA. So this bovine prolactin mRNA and raw sarcoma virus genomic RNA, they have used this in a petedosis, and that means cell culture, and found this methylation process at M6A sites. Okay, so this methyl transferase complex mediated methylation has been observed both in vitro and in vivo. Okay, so this is all about N6 methyl adenosine. Okay, we are going to discuss about it more and more. So this is the answer of this 5 question. Fine, let's move to next question. The differential M6A profile is the hallmark of heart disease, liver disease, cancer or non. So what is M6A? That is your N6 methylation of mRNA. Fine, this is the short form of this long term, that is M6A. Yes, answer this question. very good. Like you answered correctly. So this M6A profile is the hallmark of cancer. Okay. So what is it? The differential M6A profile is an emerging hallmark of cancer. Aberrant M6A deposition plays a key role in tumorigenesis in various cancers by broadly altering the gene expression. So aberrant M6A deposition, what is the meaning of it? M6A deposition means when your adenosine nucleotides gets methylated at the sixth position, okay, and this happens a lot of times, so that obviously it will just alter the gene expression. Why it will alter the gene expression? Because after the M6A, that is mRNA, gets modified, it will produce your malfunction proteins. Okay, so in that case the person will face different diseases or syndromes. M6A methylation plays an important role in wide variety of biological and pathological, including cancer development. This MET-L3, M6A methyl transferase. So this is the enzyme, okay, MET-L3. So what is the methyl group donor? That is your SAM, S-adenosyl methionine. It produced as a byproduct of liver. Okay, it produced from liver. Is upregulated in gastric, colorectal and liver cancer. It is highly upregulated and it has been found specifically in gastric, colorectal and liver cancer. So what is the function of this enzyme? This adds the, as the name suggests, this adds the methyl group to the adenine nucleotide at the sixth position. Okay. So M6A modifications frequently involve key components of oncogenic pathways like Wnt beta-catenin pathway, PI3K, AKT and mTOR signaling pathways. You can go and read about all these pathways if you are interested, otherwise it will be too long. Okay, eventually contributing to their aberrant activation of cancer. Understanding the function and molecular mechanism of M6A mediated epitranscriptome may thus unravel novel therapeutic targets and biomarkers for cancer detection. Why we need biomarkers? Generally it happens that cancer requires in sometimes in, if you heard about it, like from your society or from other people, or in case of research also you have studied that cancer, when a patient goes to the doctor and find that there are some abnormalities been happening and some issues he or she been facing, so at the end if the reports comes, the doctor says that this is the last stage of cancer, or this is the third stage or second stage of cancer, you have very little time left. So why does this happen? Because of your late diagnosis. Cancer is a thing which cannot be diagnosed from the early phase, and specifically your gastric cancer, colorectal cancer, liver cancer, they just spread out so fast using through the metastasis process. What is metastasis? So in case of metastasis, one cancer cell flows out through the blood to add from one position of that organism to different organ system, and they produce cancerous cell at that particular part. Okay, like for example, if your some person have cancerous cell in the liver, it will, some cancer cells will get floated to your stomach and they produce the cancerous cell or tumor in stomach. So in that case this is called metastasis. Okay, and when metastasis happen, this is so fast, this spread so widely that it is very but difficult to cure it completely. Okay, so that's why it is very important to get a biomarker so that we can detect or diagnose the cancer from a early phase, so that a person can be recovered. Fine. And there are several novel therapeutics targets and the research have been going on, so if you want you can contribute it also. Fine. So this is about M6A methylation. Next is factors that cause an RNA modification is called readers, writers, eraser or modifiers. Which one is it? Let's see. What are these readers and writers? So these M6A regulators, they have been classified into three types. That one is writer, eraser and reader. So what is it? More recent studies have characterized other key components of M6A methyl transferase complex in mammals. So writers including MET-L14 or WTAP, or for example this MET-L5. The hypothesis that M6A in mRNA being dynamic and reversible is proven with the discovery of first M6A demethylase fat mass and obesity associated protein FTO, that is called eraser. Another M6A demethylase ALK-B homolog 5 was later discovered as well. The biological function of M6A are mediated through a group of RNA binding proteins that specifically recognize the methylated adenosine on RNA, that is called reader. These binding proteins are named M6A readers. So what is it? What is reader, what is writer and what is eraser? First thing, as the name suggests, reader means it recognizes or identifies the M6A methylation. For example, if the mRNA have some M6A methylation, that means your sixth position of your adenine nucleotide get methylated because of some reason. So there are some M6A readers which recognize it. Okay, we need to first identify where is the methylation. Fine. Next is your eraser. So what is the eraser? Erasers are called demethylases. So as the name suggests, demethylase means it will just remove the methyl group. Okay, so this removal of methyl group happen by demethylase enzyme, and example inside your body is FTO protein and ALK-B homolog 5 protein. So these are the examples of erasers. Understood? Next is your writer. What is the writer? Writer means those enzymes are called methyl transferase. So what is it? So this methyl transferase enzymes, they modifies the sixth position of adenine and add a methyl group there. That means they just methylate the adenine molecule, adenine nucleotide. That's why it is called writer. What is the example of writer? It can be MET-L14, MET-L5, and these are different examples have been given here. Understood? So now you understand the whole mechanism of M6A regulator, which can be classified as writer, which modifies the adenine nucleotide and add a methyl group, or just do the methylation process. Eraser are the demethylase enzymes which remove the methyl group. Okay, that means it just clears out. The third one is the reader. Readers are those enzymes which identify, okay, those recognize the position where methylation has been happened. Fine. So the readers are very important. As they recognize the position, then eraser comes to that place and remove the methyl group. So what is your answer? Factors that cause an RNA modification or add a methyl group will be your methyl transferase enzyme, that is your writers. Readers are those which recognizes, erasers are removal of methyl group. Fine. Let's move to next question. So YTH domain proteins are considered as RNA modifiers, epitranscriptome writers, epitranscriptome readers, or epitranscriptome suppressor. What is it? Yes, answer this question. Okay, let's see. As you have said, C. So fine. So this YTH domain, the YT521B homology, that is the short one is YTH domain, family of proteins, and these are different isoforms, have been characterized as direct M6A readers. Readers means what? They recognize the position where your methylation of adenine nucleotide happened. Fine. And have a conserved M6A binding pocket. So this is conserved. Insulin-like growth factor 2, mRNA binding proteins like 1, 2, 3, like short form is IZF2BP123, are reported as novel class of M6A readers. So these are good examples of M6A readers. I think you found some examples of erasers like FTO and ALKBH5. Writers are like MET-L14 or MET-L5. So here is an example of your reader. Okay, and IZ2BPs use the K homology, that is KH domains, to selectively recognize M6A containing RNA and promote the translation and stability. Okay. So next is these M6A readers together with the M6A methyl transferase. This methyl transferase is what? These are the writers, and demethylases are what? They are the erasers. That means they remove the methyl group. That's why they are called erasers. So some modification was done and they remove the modification. So they are called erasers. Establish a complex mechanism of M6A regulation in which writers and erasers determine the distribution of M6A on RNA, whereas readers mediate M6A dependent functions. M6A has also been shown to mediate structural switch termed M6A switch. So you can go for studying about more M6A switch and all. So this is a brief introduction about your N6 methyl adenosine and how this interacts and what are the different types of regulators of M6A adenosine methylation, that is readers, writers and erasers. Okay, and there are some examples are here. You can just remember one or two examples of each case for readers and writers and your erasers. Okay, this will be helpful for understand doing some MCQ questions when if it comes to your in your exam. Okay, I think it is clear to everyone. If you have any question regarding this, you can ask me. So these are obviously readers. Next is this from here onwards we are talking about translation process. Fine. So mRNA codons are usually discontinuous manner, distinct manner, overlapping manner, three frames manner. Which one is it? So this is all basic things about translation process. As the RNA molecules are involved and regulates the translation process, here we are talking about the different types of translation machineries. as you have said B, so this is your correct answer, but we are going to discuss few things about mRNA codons. So the three roles of RNA in translation. Protein translation by ribosomes require three types of RNA. The messenger RNA specifies the amino acid sequence of a protein. Where comes the messenger RNA? And always remember this messenger RNA, if we are talking about your eukaryotes, this messenger RNA undergoes post-transcriptional modification before moving towards translation process. Fine. So this nascent mRNA, which just produced from the transcription machinery, this undergoes post-transcriptional modification where the splicing happens. That means removal of introns happen. Okay, the non-coding parts got removed. Second thing happen, your capping. Okay, capping, and other one is adenosine tailing process. So this capping, tailing and splicing mechanism, these three are called post-transcriptional modification. And after that, this mature mRNA will move to the cytoplasm, and there only your translation machinery will move on forward. Okay, but if we are talking about prokaryotes, so prokaryotes, this doesn't happen, this post-transcriptional modification doesn't happen in prokaryotes. There happens only as the mRNA produces from the transcription process. Suddenly, just a seconds after that, the translation machinery starts. Okay, that's why it is called transcription and translation are coupled mechanism in case of prokaryotes, but not in eukaryotes. This is one good example or good differentiation between prokaryotes and eukaryotes if someone asks you. Fine. So mRNA, as we know, this is the messenger RNA and it has all the codons and all the sequences. And each amino acid is selected based on the order of triplet codon. Why is it triplet? Okay, we are talking about it. Next, the transfer RNA. Fine. So it converts the information in mRNA codons into amino acid sequence of the protein. And the transfer RNAs, they carry the amino acids specified by the codons and base pair with the codons by their anticodons. So where is the anticodon present? In the mRNA or tRNA? Where? Anticodons are present on the tRNA, okay, and codons are present on mRNA. Always remember this. Some people get confused between anticodons and codons. So anticodons are those residues which are present on the tRNA, and codons are those which present on the mRNA. Okay, and ribosomal RNA makes up the bulk of the mass of ribosome. One rRNA species, that is 28S RNA, is a ribozyme that catalyzes the reaction in which the peptide bond is formed. Fine. So if you see here, this is your mRNA from 5' to 3' direction, and you find out that this is your tRNA with having some anticodons. So this triple U, these are anticodons. But here you find these are the codons. Okay, these things like GGG, AA, they are the codons. And from this tRNA acceptor arm, it binds to the amino acid of choice. Like for example, this U, they form the hydrogen bond with your AAA. After that, the tRNA molecule will get the amino acid which specified for this codon only. Okay, and this is the direction of movement of ribosome, 5' to 3' direction. So this is about genetic code. So the codons for 20 standard amino acids are specified by triplets based on the known as genetic codes. Why is it triplet? For example, you say that because there are 4 to the power 3 equal to 64 possible combination of triplet codons are there, and most amino acids are specified by more than one codon, that is degeneracy of the codon. What is degeneracy and why there are 64 codons are available? We have 20 standard amino acid and we have 4 nucleotides. Okay, adenine, thymine, guanosine and cytosine. Okay, so 4 nucleotides are there. if we go for one codon, like 4 to the power 1 equal to 4, that's why it will produce or it will go for only 4 amino acids. For example, we can go for 2. Okay, that means 2 bases like GC, GU or AT, this will stand for 1 amino acid. If we go like this, then 4 to the power 2 equal to 16 amino acids can be possible. So what about the rest 4 amino acids? Fine. Next, move towards 4 to the power 3. So this will be 64. So 4 to the power 3 means 3 letters, 3 residues like ATC, GCC or AUG. So that means 3 nucleotides combined to produce or to stands for 1 amino acid. So in that case we got 4 to the power 3 equal to 64. So but we do not have 64 amino acids, we have only 20 amino acids. So what happens then? That means one amino acid can go for 3 to 4 triplet codons. That is called degeneracy. Okay, like example of your lysine. Okay, this lysine amino acid can go for 3 to 4 amino acid codons. Fine. So this will be called degeneracy of codes. Understood? And 61 codons specify the amino acid, and there are 3 stop codons already present. What are the stop codons? UAG. Everyone knows this, but still I want you to remember and clear about it. UZA and UAA. Fine. So these are 3 are called stop codons. So when these codons arrive in the mRNA sequence, the protein formation or the translation process halts there, and methionine is the start codon, AUG. Fine. So this is all about your codons. Understood? So methionine stands is the start codon, which is AUG. Okay. Now how the triplet code got read? So there are 3 potential read frames in all mRNAs, and always remember they read in a non-overlapping manner and comma-less manner. That means there is no comma in between any triplet codon, and they are non-overlapping. Rarely are mRNAs read in more than one frame. Likewise, frame shifting is very uncommon. Okay, and what is frame shifting? Let's see. For example, here this is the sequence of codons present on the mRNA, and your ribosome or tRNA molecule is going to read it like GCU stands for alanine, next UGU, and it will move like this. So it will be the amino acid or polypeptide sequence. If we just leave the G molecule, that is your guanosine, if we just leave the G, for example, in some cases I just deleted the guanosine from the starting frame. Okay, now what will happen? Now the frame shifts. How does it shift? Like see, at first it was GCU, now it is CUU, GUU, and for example like this, it will leave the only adenine group, and you can see here that all the amino acids got changed. Okay, now for example in the cell I want this particular protein to function a metabolic process, but because of the deletion of guanine I am getting another protein. That means this metabolic process will not going to happen, and this is the malfunction protein. This protein can lead to different diseases also. So this is one example of your frame shift mutation. Okay, when there is a single mutation of your nucleotide happen, single mutation or single insertion or deletion happen, which lead to shift the frame. Here you can see that the frame got shifted. So if the frame got shifted, that is called frame shift mutation. Okay, understood? So I think it is clear to everyone. And for example, if your stop codon arises, okay, that is called nonsense mutation. Fine. What is silent mutation? When for example this A got mutated to C. Now it is, what will be called it, like C, G and C. So this CGC will stand for the same amino acid. Why? Because this is called obol base. Okay, this is called obol base. Because of the changes of this a single base, the name of amino acid which it codes for will not change. Understood? So this is all about triplet code and your degeneracy of code and frame shift mutation. Fine. So let's move to next question. Before participation in protein synthesis, the tRNA must get modified, denatured, amino acylated or acetylated. Which one is it? Okay, let's see. So this should be amino acylated as you have answered. So why it should be amino acylated? Let's look into the structure of tRNA at first. Okay, tRNAs are typically, transfer RNAs are typically 70 to 80 nucleotides long. They all have a clover leaf like secondary structure and fold into a L-shaped tertiary structure. Okay, for 4 double helical stems occur and 3 of these have loops of 7 to 8 residues at their ends. One loop, the anticodon loop, this is called the anticodon loop, which interacts with your codons present on the mRNA. And the upper stem is called the acceptor arm. This is your acceptor arm. As you can see, this is has a 3' residue. Why the 3' arm, 3' end of the RNA is acceptor, not the 5'? So why is it called 5' and 3'? First thing is, there is the 5' phosphate group present at the sugar molecule. Okay, so we know that nucleotides are made up of 3 parts: ribose, sugar, next is your base, nitrogenous base, and phosphate group, right? So there is one phosphate group present at the 5' position. That's why it is called 5' group. When there is a phosphate group, so that other molecules cannot get added. But when we are talking about 3' group, that means this is called 3' OH group. Okay, this is basically 3' OH group. So as you can see, it has a hydroxyl group, so that it can form hydrogen, it can form bond, hydrogen bond or whatever it is. It can form a bond with different molecules which can get attached to it. Okay, and this is the 3' position where this amino acylation happen. Also, in case of DNA replication and RNA transcription, we say that all the nucleotides get added to 3 days end of the template, not the 5 days. Why this 3 days end? Because the 3 days end, it has open OH group or hydroxyl group, so that the nucleotides can get added and form the phosphodiester bond. But when there is a 5 days end, it cannot form a phosphodiester bond there. Understood? Or you can say that nucleotides cannot get added. Fine. Okay. So the upper stem is called the acceptor arm and ends with a CCA end. The amino acid is attached in ester linkage to the 2 days or 3 days OH of the amino acid A residue. Many residues are modified in tRNA and some modifications are shown in figure. So this is the tertiary structure which we can see in a 3-dimensional manner, and this is a 2D figure. Okay, so this is your dihydrouridine and everything got. These are some modifications here we can observe. So this is the T schedule, this is D loop, and this is your anticodon, and this is your acceptor arm. So two-step process for mRNA decoding. So tRNA specifically go for this mRNA decoding process using the interaction between codon and anticodon. How does it happen? Amino acids are attached in ester linkage to the 3 days terminus of tRNA, forming amino acyl tRNA. Okay, and the enzymes that carry out these ATP driven reactions are called amino acyl tRNA synthetase. Synthetases are those which synthesis something. Okay, there is some product will get produced, and amino acyl tRNA synthetases are highly accurate, that means high fidelity, and helps in minimize the translation errors. Next thing, what happens? This amino acid is added to the growing protein chain based on the codon-anticodon interaction between mRNA and tRNA. Here you can see amino acyl tRNA synthetase specific for phenylalanine. Okay, this is your amino acyl tRNA synthetase, and this is tRNA specific for phenylalanine. So how does this happen? We just say that the acceptor arms carries the amino acid. How does it carry the amino acid? By forming a bond or linkage. Okay, so linkage of phenylalanine to tRNA happens using one ATP molecule. That means it should this is the energetic process. Energy will be included, or energy will be expended. So, and here you can find out this is the COOH group, okay, of phenylalanine, and this is the OH group of your 3 days chain. Fine. So now you can see a high energy ester bond got formed between COO after removal of one H2. Fine. This is COH carboxyl group, OH hydroxyl group. After they interacting, they produce a high energy ester bond. Fine. Next, this amino acyl tRNA binds to the UUU codon. Net result is phenylalanine is selected by its codon. Understood how this amino acyl tRNA just brings one amino acid? We generally say that tRNAs are transfer RNAs, they just bring the amino acid. How does this happen? By forming a high energy ester bond. Okay. Next is bacteria synthesizes 30 to 40 tRNAs, whereas the eukaryotes may synthesize 50 to 100 mtRNAs, and they all are specific towards the specific amino acid. Okay, the 20 amino acids are there, so they will be specific towards the respective amino acid. Thus, the given amino acid often can be carried by more than one species of tRNA. Got it? So that means, for example, I can call that lysine or arginine. Like for example, lysine. So lysine can be carried by 2 or 3 tRNA. Okay, not a single tRNA. So each amino acyl tRNA synthetase recognizes one amino acid and all of its cognate tRNAs. Understood? So why this amino acyl tRNA synthetase is important? Because this interacts with the OH group to form a bond. Fine? Fine. Let's move to next question. Bacterial mRNA codons are not similar to plant codons, not similar to virus codons, similar to other animal codons, or similar to mitochondrial codons. Which one is it? as you said, some of you said C or some of you said D. So this is your answer, similar to animal codons. Why is it similar to animal codons? Always remember this thing, that genetic codons are universal. Universal means just a few exceptions are there, but they all are similar in case of your bacteria, virus, plant and animal. All the In all the cases like bacteria, virus and all the organisms, these genetic codes are similar. They are universal, okay, with few exceptions. Sometimes some organism have some exception, okay, that's why it is similar to animal codons. Next, here you have some 3 to 4 true and false questions, you can answer it, okay. Protein synthesis can happen only from mRNA, is it true or false? Okay, so as you said, it is true, because it can happen only from mRNA. This is the messenger RNA which produced from transcription process, and it has all the genetic information in it. But in case of tRNA, we get the secondary structure, and from secondary structure we cannot produce a chain of protein. Okay, and also it doesn't have that much genetic information as mRNA. Fine. And rRNA specifically found for ribosomal RNA synthesis. All mutations are detected and eliminated from cells. Is it true or false? Okay, as you said, it is false. Okay. All mutations cannot be detected and eliminated from cells. There are different ways of detection and elimination process, as we just talked about different types of modification, RNA modification, RNA regulators like readers, erasers and writers like this. So all cannot be eliminated from cells. So low mental or physical stress maintain long telomeres. Is it true or false? is true. As you have already discussed about some experimental results from published in Journal of Science, that if a person in like in case of athletes or young athletes, the telomerase activity just increases upon exercising. Also, in case of middle-aged athletes, the telomerase activity increases by 1.9 fold by continuous exercise and all. So that's why it is always said, and it has been scientifically proven, that low mental or physical stress, that low stress should be better to good for your health. Okay, like doctors always say that you should stress less, and this low and mental physical stress can increase the maintenance of long telomeres. And this long telomeres will, what will happen, this long telomeres will just keep on increasing by the telomerase enzyme, and that's why the cell, uh, the age of the cell will increase. Like if it is generally go for 20 years, then it should go, then it will be go for like 20 to 25 years. Okay, so this is the final question of today's session. Adequate vitamin C in diet ensure longer telomere length. Is it true or false? Okay, as you said that this is true. Okay, adequate vitamin C in diet ensure longer telomere length. That means it increases your cell's lifespan. And there are few experimentation results have been shown in the lecture videos, that how vitamin C can increase the telomere length, and it has been really got interesting if you see the results. Okay, so you, if you want to know more about it, you can go for the lecture videos. Fine. So today we discussed about different types of RNA modification, like N6 methyl modification, including the modification regulators like readers, writers, and erasers. Okay. Before that, we just talked about telomerase enzyme, and this is a TS65. This is a molecule which have been extracted from a natural Chinese herb called astragalus. And also we talked about translation process, how this mRNA, tRNA, and rRNA molecules have been combinedly affect the translation process or translation mechanism. Okay, still if you have any question regarding this, then you can ask me, so that I can clear it out. Okay guys, thank you for joining the class. And I think we have a extra session for RNA biology. So if you get the link, then you can join, so that you can just brief about all the things what we have studied from week 1 to 12, and it will be a overall session for everyone. Okay, see you then.